Hi, everyone. Welcome to the very last session, but absolutely the most important session of CHS 2024 about species at risk. We're not sure why this talk is in the species at risk section, but Rachel will tell you all about it.
Okay, awesome. Hi, everyone. My name is Rachel Dillon, and I'm currently the research manager for Christina Davy's Wildlife Conservation Lab at Carleton University. Thanks so much for coming to listen to me talk about best practices for replicable surveillance of Ophidiomyces, ophidiicola, which I'm going to call Ophidiomyces from here on out. So this was developed in response to an ECCC contract in collaboration with Aaron Allen and Christina Davy at Carleton. And then hopefully we're going to get to our big call to action at the end of this, where we're hoping all of you guys, whoever swabs snake or whoever hangs out with snakes in the field, could maybe do some swabbing for us. So first, I'd just like to thank our project partners and collaborators, including Environment Climate Change Canada, with a special shout out to Mike Asimov and Cynthia Pekarik and the Herp Health Working Group, who without their help and collaboration, this would not have happened. Also, a big shout out to the Canadian Wildlife Health Herping Group, the Canadian Wildlife Health Cooperative, and Lenny Shuros, who's there. And lastly, the folks in our lab on the Wetlands and Reptiles project for your continued support. So I also realized that I'm in the species at risk section, but I just wanted to clarify at the top that Ophidiomyces affects all snakes, not just those that are at risk, and does not seem to be causing snakes to become at risk. So just a heads up front. Awesome. So let's start off at basically the very beginning. How do you get disease? So in no particular order, you're going to need an infectious pathogen. This could be a bacteria, a virus, or a fungus. And then you need a conducive environment for this infectious pathogen to thrive. So for example, this could be a marsh or a first grade classroom. Lastly, you need a susceptible host, like this wild eastern fox snake. And each of these, pathogen, environment, and host, must all be present in order for a disease to occur. The disease triangle, if you will. So when we're looking at this in terms of Ophidiomyces, or as some of you may know, snake fungal disease, we're looking at it in terms of the fungus, Ophidiomyces ophidiicola, and that's the fungus that causes Ophidiomyces. So cases of Ophidiomyces and the pathogen have been documented worldwide in a variety of habitats, both captive and wild snake populations. Ophidiomyces may cause lesions or other clinical signs of disease, and in some cases it can cause death. However, this is not always the case, as some snakes that carry the pathogen never exhibit lesions at all, or are able to resolve their lesions over their active season. Okay, so there's three things that must occur for a confirmed Ophidiomyces diagnosis. First, a snake is showing clinical signs of Ophidiomyces. These physical signs are highly variable and sometimes include lesions, crusty scales, pustules, and or emaciation of the snake. In some cases, like I said, it can cause mortality. Second, when analyzed via qPCR, a skin swab is positive for Ophidiomyces. Last thing you need for a confirmed Ophidiomyces diagnosis is the suspected, oh my gosh. Lastly, what you need is to do a histological analysis for fungal elements of the biopsy skin that you have to take. So you're looking for the actual fungal elements under the slide at the point. So with all three of those things in place, you can say that a particular individual has Ophidiomyces. With only clinical signs and a positive body swab, so these first two items here in pink, you just have suspect Ophidiomyces. The histological analysis is needed to confirm full disease. So, with all that being said, is Ophidiomyces a conservation concern? Based on current evidence, no. Studies have found that when looking at peer-reviewed literature in which samples were tested for Ophidiomyces or in which gross lesions were reported, they did not find that there was an increase in instances of lesions or samples that were positive since 2008. In addition, we have also found evidence that suggests that many, if not most snakes, are able to resolve their lesions and clear the fungal pathogen during their active season. So, although individual snakes with Ophidiomyces do sometimes die, the data available from the United States and from opportunistic sampling across Southern Ontario do not suggest high mortality rates. It's known that Ophidiomyces is present across southern Ontario, but suitable data to confidently estimate distribution limits or prevalence of the pathogen do not yet exist. And this is where pathogen surveillance comes in. So this only requires that skin swab part of that 3-step processes that I showed you previously. So just the skin swab. So to conduct ophidiomycosis surveillance, you can use skin swabbing protocols in the field over a certain geographic region to provide you with presence-absence data of the fungal pathogen. We were first asked by ECCC to summarize swab sampling protocols during fieldwork based on a review of publications on ophidiomycosis to date and identify sources of potential variation, which then inform Canadian surveillance guidelines. We pulled sampling, storage, and analysis methodology from 102 peer-reviewed papers that conducted samplings of snakes for ophidiomyces. Okay, so from this, we summarize the factors that could alter detection probabilities among studies. There were high levels of variation throughout snake swabbing processes, but here are just a few examples that I'll go into a little bit more detail. So if all this variation in methodology can cause variation both in the detection probability of the fungal pathogen, in addition not being able to compare studies against each other from different geographic locations. So variation. So if you look at the first pie chart over here in the planning stage, so like when you're getting ready to go out and swab your snakes, one of the questions that may come up is, what swab material will you use? You can see here that there's many different materials that were used, such as cotton, polyester, et cetera, et cetera, et cetera, and all of these can impact your DNA extraction. So in the second Venn diagram here, or Venn diagram pie chart here, on the at the point where you're sampling the snake, where on the snake's body will you be collecting your sample from? Did you swab the whole snake? Did you swab just the lesions? This can also influence the fungal load that you're picking up on your actual swab. At the DNA extraction and qPCR analysis stage, what will your cyclic threshold look like? So you can see here, there was a big variety of cyclic thresholds. So this is basically your cutoff point for what you decide is going to be a positive sample for Ophidiomyces. And usually these are produced in the lab that is doing the qPCR analysis, but it's very variable across the range, and most times it wasn't even reported. And this can definitely influence whether, whether or not your complete picture of presence or absence of the disease. So from this literature review, we were then asked by ECCC with input from the Herp Health Working Group to develop sampling protocols for Ophidiomyces surveillance that are 1, repeatable, 2, feasible from a field collection standpoint, and 3, increases the probability of fungal yield of Ophidiomyces. We created infographics which outlines details for each steps of the planning process, which includes, number 1, planning recommendations, 2, swab collection recommendations, and 3/4, DNA extraction and qPCR analysis recommendations. And from those, we made the following recommendations. So first, when you're planning to swab a snake, at what point of the year will you be doing this at? Ophidiomyces is the most prevalent. Ophidiomyces, is the most prevalent directly after emergence from overwintering. So in Canada, our snakes have. Oops, I went too far. In Canada, our snakes have that overwintering period where they tend to be underground, generally from the months of October to April, or hibernating, and that restricts our sampling season to the spring and summer months. Recent studies have shown that disease becomes more severe during that hibernation period, and that there's higher prevalence of both clinical signs and pathogen on the snake post-emergence. The detection probability is therefore highest in the spring, giving an accurate picture of highest pathogen probability, and the snakes may be more concentrated in one area at this point. So therefore, we're suggesting that we conduct surveillance for Ophidiomyces in Canada in the spring months, right after they're emerging from their overwintering period. So next, we made recommendations on the types of materials that you should be using in the field. I'll just touch on one of them here. So we say most studies have used cotton swabs, but we've actually found, and other studies have as well, that there's a higher yield of DNA for extraction with the polyestrine swabs. So therefore, we're going to make the recommendation to use these polyester tipped swabs applicators, and this will hopefully reduce variation amongst studies. Okay, and lastly, when conducting disease surveillance, definitely decontamination methods should be employed. And we recommend that we follow the decontamination protocol from the Canadian Wildlife Health Cooperative, who have already laid all the steps out, including bleaching your equipment and changing gloves in between individuals and whatnot. So I won't go too much into this swabbing guideline here, but because it is all available in our guidelines that we will that you can ask me for afterwards. But basically, this is our sampling kind of schematic. When you encounter a snake in the field, if it has lesions, you follow a certain protocol. If it does not have lesions, you follow a different protocol. Basically, you're ending up with your samples at the end here, though. The important part of this, though, is that is in the field storage and in the lab storage. So basically, our biggest problem is the freeze-thaw cycle in terms of losing DNA. So you want to get those samples into a cold place as fast as possible. Ideally, your -20 or -80 freezer in the lab. And lastly, we've got our DNA extraction recommendations and QPR analysis recommendations. Won't go get into the nitty-gritty of this too much because I don't know much about it myself. I'm not the lab person that does it. We send a lot of our samples off for other folks to do it, but we recommend using this particular kit, the Keogen kit, as well as the qPCR protocol that Allender outlined. And lastly, that brings me to my call to action. So with the recommended surveillance guidelines in place, we now want to move into the next stage of the project, which is a cross-Canada ophidiomycosis surveillance blitz, hopefully coming in the spring 2025. We are currently working on logistics and gathering funding for sample processing, but in the meantime, we'd like to put out our call to action. So we're looking for folks who already work with snakes or come into contact with them frequently during their own fieldwork. And in particular, we're looking for people who work with snakes in Quebec and then the more northern regions of provinces. We've got a lot of that south of the border kind of filled. So with that, I'd like to say thank you. And if you have any questions, please feel free to reach out to myself or Christina. And if you're interested in swabbing for us, also do that. Thank you.
Great presentation. I was just wondering if you know anything about if snakes are able to detect individuals that have SFD. You said it was like more prevalent in their overwintering, and if they would be able to like sense that and maybe be more particular about what sites they're choosing based on that.
Good question. There's definitely a lot of work being done in terms of what habitats are associated with the prevalence of disease, as well as we found snakes actually hibernating or in their brumation. And, oh, yeah, sure. Sorry, thank you. Yeah, the fungus is kind of ubiquitous about the environment and everything. So I'm not sure if they're actively like avoiding areas knowing that the fungus is there, but there definitely seems to be something more going on with the transmission for sure. Thank you. Thank you.
So next in our session, Ryan Wolfe is going to talk to us about understanding trends in abundance, survival, and behavior of Canada's only population of blue racer Coluber constrictor foxii.
Thanks. Round two. Thanks. So if you didn't see my last talk this morning or a little bit ago, my name is Ryan Wolfe. I am a biologist at Natural Resource Solutions. And in the last session, Briar said she's obsessed with frogs. I'm obsessed with large colubrids and specifically racers. So I've had the utmost privilege of working with the last remaining population of blue racer in Canada for the last, I guess, 7 years now, since 2018. And so I'm going to be talking just about a little portion of what I've been doing. And it's kind of an update on a talk I did a few years ago at CHS with, where did the pointer go? Oh, there it is. With regards to population estimates, which I started as a master's project in 2020. Okay, so yeah, like I said, we're going to talk about population estimates, get into analyzing some abundance trends, and then also hopefully some exciting finds and stuff if we have time. So if you don't know about the species, or I guess the subspecies, I should say, they are a very large, very fast terrestrial snake, and also very beautiful, if I can say so myself. They are prey generalists that occupy open canopy habitats. And most notably for us, we only have one single population, and that is on Peely Island in Ontario. So the reason for doing a population estimate, first and foremost, is we want to know how many snakes are actually around so we can inform conservation decisions. And also so we have an idea of how urgently those conservation actions may need to be implemented for the species. And another reason I started doing this one around now is because we didn't know what the population has been at since about 2002 when the last census was done. And they predicted that there was only about 140 snakes remaining at that time, and the population was severely declining, so much so that they hypothesized that they would be extirpated by now. So, of course, we started doing the mark recapture surveys to assess this, and we also were able to add in more sites that they didn't have access to back then, and update some of the models. So we did our surveys across 4 different sites within the known range of blue racer. We labeled those sites A through D, and we conducted surveys for 5 weeks every spring and 2 to 3 weeks every fall, using a free search or visual search method. And all snakes were captured by hand. Individuals that weighed more than 30 grams, which is pretty little, were pit tagged so we could identify them as recaptures if they were caught again. And then all of the capture histories that we got from these individuals were put into models. So when I designed this project with Dr. Niall Rawlinson at the University of Toronto for my master's work, we designed it to be implemented with a robust design model. So it has an open and closed component of the population. The primary sampling periods are the seasons, so springs and falls, and those are considered to be open. And then the closed are the weeks within those primary sampling periods. So I created a series of 24 different models that had different effects of time and primary sampling occasion, as well as design covariates of survey effort and season operating on the capture and survival probabilities for the individuals within the model. And then I took the top models that made up the 95% of the weight of those 24 to do the model averaging to produce the results. So before we get into the actual results of how many snakes we have, just kind of a really brief overview of the capture numbers we're dealing with. I couldn't produce good population estimates early on because we had such sparse data. Back to 2000, 2002, 20 years ago, they had marked 106 snakes over their 3 years and had 15 captures over that time frame. And now we, over 4.5 years, have been able to capture 252 individuals, and 280 of those are recaptures. Now those recaptures do include every week rather than every year like they had, but so now we finally have enough data to start producing accurate estimates. So the top models had effects of season operating on survival and season and effort operating on the detection probabilities. And what was interesting and surprising was that the models predicted that survival was actually highest over the winter, which is the opposite of what is reported in papers, but it was near perfect. And then the survival over the summers was quite a bit lower at 0.68. Okay, so the robust design produces a abundance estimate for every season, every primary sampling period. And as you can see in the first few there, the confidence intervals were super large. And I ended up actually just getting rid of the entire first year, the spring and fall, not because the confidence intervals were large, but because in the spring we didn't have access to all of the sites that we were sampling over the rest of the time period. And in the fall, we had an extremely reduced sampling period and a resulted very, very low number of snakes, which is why those confidence intervals are so high. So when we get rid of those, the trend goes from so slightly increasing to very slightly decreasing. But for the majority, what we found, which is really exciting, is that over the last 4 years, it's actually a pretty stable population at about 142 individuals. So it's a low number, but it's definitely not extirpated, which is really good news. And so then I wanted to look at trends in abundance, looking at how many they had back 20 years ago and how many we have left now. And to do this, I had to remove our Site D and our fall sampling periods because they didn't have access to Site D back then. And I can't say where that site is because of obvious reasons. They're extremely endangered. But so I had to remove those and then do the estimates again for just sites A, B, and C. So a reminder, they had about 140 snakes during that period. And they also did an estimate for one site specifically, site B, which they expected to be the, stronghold or the last remaining hope for the species. It's where they caught the most of their snakes, and they had estimated that to be 79 individuals. So looking at sites A through C, our abundance appears to have a positive trend, which is fantastic, but we have lost about 25% of the snakes there. So we've gone down from 141 to 107. And then if we look at site B specifically, we've had an even bigger decrease where we've lost 40% of those snakes. We've gone down from 79 to 48. But there still is like a slightly positive trend, which could be a good thing that they're at least not declining further within the last 5 years. So one thing I also wanted to mention is there was a potential mortality event in winter of 2021 to 2022. But if you look at this population estimate there for spring of 2022, we actually have one of our highest estimates. And so I was really curious of why that would happen, why it wasn't reflected, what we were observing in the field, why that wasn't reflected in the models. So when I looked at the actual demographic characteristics of the capture data that we had, you can see that the number of adult individuals that we capture per spring at this site specifically usually hovers around 12, 13 individuals. But in 2022, we went all the way down to only 4. We've never got those individuals back. But on the flip side, the juveniles, which we have usually very, very few of, we actually had 12 in 2022. So what I'm thinking has happened is because the racers in our selected species, they grow really fast, reproduce really quickly, those individuals would have been adults by the following year, by the spring of 2023. So they may have already effectively replaced those individuals. It's not that we got those adults that we had before back, it's that they've been replaced already, which is super exciting in terms of like a recovery standpoint from population events. Okay, so have a little bit of time left. So going to go into some of the exciting finds that we've had over the mark recapture study. One is a potential range expansion, which was not expected, but the current range is highlighted in yellow on that map. Since 2021, we have had several observations from the extreme western side of the island where there hadn't been many reports since about the 70s. There had been a few in a little bit on the western side of the edge there from Ben Borchuk's radiotelemetry work in the mid-90s, but for the most part, they were restricted there, and we've had potential even overwintering on the extreme west side of the island. So it's super exciting, and more work needs to be done there to figure out if they are actually occupying that side a bit more. Another super exciting find is that we've found 2 new potential hibernacula. They fit all the same characteristics of the few known ones that we have, the large hibernation complexes. And so this year and next, we're going to be exploring those to see if we can't add a few more sites to future work. And it would be super important for protection for the species as well. Okay, so another thing that we've noticed is we've had behavioral shifts in the activity of blue racers on Peely. We started to notice that they were changing their basking behaviors. Peely Island has become overrun by shrub, and for a terrestrial species, that could be a problem. But rather than necessarily seeing them completely absent from all sites like hibernacula, when we do our surveys, we've started finding the snakes actually to be aerial basking rather than terrestrial. So on the left, you can see a snake. It's hard to see, but it's just lined up right there. That's in the sun. There's still vegetation around it. It could climb if it wanted to, but it prefers to be on the ground. In this extreme shrub thicket, it's basking up in the air, and we've seen up to 40% more likelihood that this occurs within areas that are have dense vegetation, especially in the fall or late spring. And they'll bask like 8 or more feet above our heads, which is pretty intense. Okay, so the other thing has positive and negative. They have shifted site use. So in areas that used to be these beautiful savannas and old fields back when they found tons of snakes there 20 years ago, they've become complete shrub thickets, and we've had up to 100% of site abandonment where we find no snakes there anymore. On the other side, we have some agricultural fields that have been restored, and we get a lot of racer activity in there. So hopefully with habitat management, there's still some success stories to be had there as well. And so future steps, we just want to continue investigating the relationship between habitat succession, management efforts, and behavioral ecology of these snakes. We're conducting a PVA next year, and we want to confirm use of those potential hibernacula as well as continue our other work. So thank you so much. And if you have a couple questions, hopefully we have time.
Awesome. Thank you, Ryan. I think we have
Great.
time for 1 or 2 questions, and then we'll move on to our next presentation.
Okay. Thanks, Ryan. That was a really good presentation. I'm just curious, what factors were causing them not to be on the western side of the island and mainly only found on the other one?
The western side of the island is kind of like the people hotspot. So the eastern 2/3 is where the majority of all the conservation lands are. And it's where the only like racer hibernacula, large hibernation complexes have been documented historically as well. And on the west side, the ferry comes there, and then that's where the most traffic is. And most of the residents are kind of congregated around there. So that's probably plays a big factor. And there's not a lot of open habitat that's left. There are potential suitable areas for hibernation, like rock fissures and stuff. But in areas that are like old quarries and still actively, used by people quite a bit, and racers do not like people. Yeah.
Yeah, question from Cheryl online. Could the perceived decrease in population be due to reduced detectability due to the brush thicket?
In the certain sites where we have, so I think you're mentioning kind of about where we've had up to 100% site abandonment, and it's definitely possible. We do survey these areas still, without trying to describe them, so we can't give away locations. But basically, some of these shrub thickets are still bordered by trails, and those used to be hotspots for the population estimate 20 years ago. And we still walk those trails, and we have never had a single racer use them, despite them having like 40, 50 observations over a few years at that exact site. So it's definitely possible that they still use the shrub thickets, and we can't access all of them, but it's definitely a real decline.
Our next speaker is Hana Thompson, who is going to tell us about the genetics of the Eastern Massasauga Rattlesnake Conservation Breeding Program.
Okay, hi everyone. My name is Hana in Cambridge, Ontario, and today I'll be telling you about some of my work looking at the genetics of the Eastern Massasauga Rattlesnake Conservation Breeding Program. So the snakes that I'm studying in this research program are from all over what is now known as Ontario, Canada, on the traditional lands of many Indigenous people, including the Mississaugas of the Credit First Nation, Wendat, Anishinaabe, and Haudenosaunee peoples, and is now home to many diverse First Nations, Inuit, and Métis people. And the Indigenous people of Turtle Island have been and continue to be stewards of this land and this species since time immemorial. And I was very lucky to grow up in Collingwood, Ontario, on the banks of beautiful Georgian Bay, just a few hours south of where these rattlesnakes can be found in the wild. So I really appreciate being able to grow up there and spend a lot of time on this land. So right now, my research focus is on conservation genetics with a focus on conservation breeding programs. So these are an ex situ conservation strategy. And ex situ just refers to animals being held in human care. So whether at a zoo or aquarium or an education facility, and ex situ populations can be beneficial for conservation in a number of ways. So they can be used for research. These populations are generally a little more accessible than some of the wild populations and maybe more used to interacting with humans. But this research can then be used to inform conservation of the species in the wild. These animals can also be used for education and training purposes. Allowing people to interact with these animals can be a really powerful way of showing them why it's so important to protect them. And I can personally attest to the value of this one. This is me, sometime in the early 2000s, getting to hold a gray rat snake and really falling in love with snakes for the first time. And ex situ populations can also be used to directly restore wild populations through things like reintroductions, head starting programs, and conservation breeding programs. So this is a picture of a loggerhead shrike at African Lion Safari, part of the conservation breeding program that's been running since the late 1990s. So conservation breeding programs, the idea is if you have a population that's struggling in the wild, we can bring some individuals into captivity and manage their breeding, and then release offspring into the wild to support a declining population or to reintroduce a population that has gone locally extinct. But one challenge of conservation breeding programs, because they tend to be a smaller population, is managing them in a way that avoids inbreeding depression. So inbreeding depression is what happens when you have mating between closely related individuals that causes a reduction in fitness, which may be due to the expression of deleterious recessive mutations. So just to explain what I mean by that, we're going to go through a little example here. So I have two individuals. One is homozygous for a gene. Obviously, in reality, this would code for some kind of protein, but we're going to pretend it codes for apple pie because that's more fun. So I have one individual who is homozygous for the functional version of this protein, and one individual who is heterozygous, so they have an allele with this deleterious recessive mutation calling for a poison apple. So if you have one copy of this gene, you're fine. You can use the other one and survive just fine. But if you were homozygous for this allele, then it would have a massive reduction in fitness. And there's tons of these deleterious recessive mutations scattered throughout everyone's genome. And for the most time, it has minimal impact because they're sort of masked in heterozygous form. But where you get problems is if you have mating between closely related individuals, because they're more likely to share some of these deleterious recessive mutations, so their offspring may express it in homozygous form and have these negative fitness impacts. So we can see from this pairing, both of their offspring are heterozygous for this gene. And so we'd expect about 25% of their offspring to be homozygous for this poison apple deleterious mutation and have negative fitness consequences. But the main thing that's important is just that when you have a lot of inbreeding, you get increased homozygosity across the genome. And in some cases, this may cause an increase in expression of these deleterious recessive mutations and subsequent reduction in fitness. And one way we can quantify this is by looking at runs of homozygosity. So here you can just think of each of these bars as being the genome of one individual. And the purple bars show segments of the genome that are completely homozygous. So runs of homozygosity. These are long segments of the genome where you don't have any heterozygous sites. They can be tens of thousands, hundreds of thousands, even millions of nucleotides in length. And we can look at the amount and the length of these runs of homozygosity, and that will tell us something about inbreeding in that population or that individual. So we can use genetic data to help inform conservation breeding programs. Traditionally, conservation breeding programs are managed using pedigrees, so they keep track of who's mated with who and who their offspring are. And then they use this data to make pairing decisions based on minimizing mean kinship. So pairing individuals who are the least related to each other as possible. However, this method, you have to make some assumptions. So when the population is first founded, it's assumed that the founders are unrelated, not inbred, and equally diverse. Of course, this is not always going to be the case, depending on your source population. It's also not always possible to strictly follow mean kinship. So for example, in the loggerhead shrike breeding program, sometimes you'll make a pairing decision based on minimizing mean relatedness, but then when you put them together, they hate each other and they will not breed. So genetic data can be used sort of alongside these pedigree-based methods to help inform management of these programs. So we can use genetic data to actually calculate genetic diversity, inbreeding, and genetic relatedness, and check some of those assumptions that were made at the beginning of the program, and fill in any gaps and errors in the stud book, or add information if new individuals are added in. And we can also use it to monitor genetic diversity and inbreeding throughout the program. So one breeding program that's incorporating genetic data in this way is the Western burrowing owl breeding program in Northern California. So this is from a paper by Bosu et al. Last year. And we're just going to quickly go through their process. So basically they start with collecting genetic data and calculating relatedness of the individuals in their breeding program. And they then created possible pairings based on minimizing relatedness between individuals in a pair. Then they looked at the location and the length of those runs of homozygosity on the genomes, and then they were able to create optimal pairings to minimize the overlap of these runs of homozygosity to hopefully kind of break them up in the next generation. So if this was the genome of their focal individual, These were two possible pairings. And if we look at the pairing of the two on the bottom, we can see that there's a lot of areas where those runs of homozygosity are overlapping. So this would maybe not be a great pairing. Whereas the other pairing, they are not overlapping. So in this case, this would be the optimal pairing. So in my project, I'm hoping to do something similar for the Eastern Massasauga rattlesnake. This is Ontario's only remaining venomous snake. It's threatened by things like habitat loss and fragmentation and persecution by humans. And you can see in the range map, the gray is showing their historical range and the blue is their current range. So there's been declines across the range, but particularly in Ontario, they're now largely restricted to two populations. One in the north, which is threatened, and then one in the south, which is endangered. So in Ontario, there's a conservation breeding program that is ongoing with the goal of starting multi-year reintroductions to help support some of these declining populations. So what I've done so far is take blood samples from 55 rattlesnakes in the conservation breeding program and extracted their DNA. In the process of doing whole genome sequencing for each individual. And then I'll be looking at genetic diversity, inbreeding, so specifically those runs of homozygosity, and calculating relatedness between individuals. So these are some very preliminary results looking at runs of homozygosity. So this is just from 8 individuals that we have sequenced so far. The remainder are currently in progress. And just to orient you on the graph, basically each row will be a different individual. And then the colored bars will represent the runs of homozygosity like I showed in the examples before. In this case, these are only runs of homozygosity that are at least 100,000 nucleotides in length or greater. On the x-axis, it shows you the location of the run of homozygosity. This is just on chromosome 1. That's as much as I've done so far. The purple individuals are captive bred and the dark green are wild caught. And I just want to say I'm not reading too much into these results yet because this is a very small sample size. There's a lot more data coming. And again, this is just from the first chromosome, so not the whole genome yet. So what we can see is there's some variation in runs of homozygosity. Some individuals really just have, little runs sort of spaced across, but some individuals like this one here have some pretty big clusters of runs of homozygosity. And when I looked at even longer runs of homozygosity, up to 1 million base pairs, this individual still had some really long ones in those areas. But we can begin to see where some pairings might be better than others using this method. So if we were looking at this focal individual, for example, we can see that they have a lot of overlapping runs of homozygosity with this individual. Maybe we should avoid pairing them. Whereas these two, they don't have as much overlapping and might be a better pairing in this case. So next steps, want to finish sequencing the rest of my snakes and complete the runs of homozygosity analysis for the whole genome. And then again, look at genetic diversity and relatedness. I'm also working with Steve Lockheed and Meg Britt at Queen's University to include some wild snakes in this analysis so we can look at some comparisons between the captive and wild population as well. And then ultimately the goal is to generate some pairing recommendations for the breeding program moving forward. So with that, I'd like to thank the vast number of people involved in some way in this project. Thank you to the Research and Conservation Department at African Lion Safari. A big thank you to the Canada Biogenome Project, who is funding the sequencing and created the reference genome. And thank you to everyone who's involved in managing the captive population and collections for the captive population, as well as on the ground at Massasauga Conservation. And thank you to the rattlesnake, of course.
Thanks for your presentation. So if you, that really cool piece of results where you're showing like these two pairs could line up, is there anything else that would make them, I guess, not a good match other than their genetics?
Yeah, so I haven't calculated relatedness for any of these pairings. So that would be another thing to look at based on both the genetic data, but also the pedigrees that they've been keeping as well. And then again, you still get the issues, whereas if you put them together, maybe they don't like each other. I also haven't checked males and females in that particular pairing, so that would also be, important, of course. Yeah, thank you.
Okay, I'm going to ask a question. Oh, no, that's even better. You're good. Go for it.
Ask a question afterwards. Sounds good.
So I know you said that like sometimes if you pair snakes, like they might not even like each other, but have there been situations where you do kind of like in vitro fertilization where you take DNA from one snake and just pair it?
You should talk to Ginger, who is doing that exact research.
On that note, come on up, Ginger, and please join me in giving Hannah another round of applause.